Both of our speakers today are from Frederick. And our first speaker is Dan McVicker. He got his PhD from the Medical College of Virginia, Virginia Commonwealth University. He's been at NCI a long time. He came here in 1990 as a postdoctoral fellow, and subsequently he accepted a position in the Laboratory of Experimental Immunology. And his big thing now is he's Deputy Program Director of the Cancer and Inflammation Program. His title, The Two Faces of Redox in Immunology. Dan.
Thank you. All right, well, thank you. What I'm going to try to do this afternoon is tell you a little bit about my experience with redox biology. I'm relatively new to redox biology, to be honest, and I am not a chemist. So it'll differ dramatically from what you'll hear from the speaker at 5 and from what you heard from Dave Wink in some of the earlier parts. So what I'm going to do is tell you about some of the interfaces between redox biology and immunology. You know, the title on the original program said immunology, which is a little bit more than a 1-hour talk. So we can't cover all of immunology. So what I'm going to try to do is show you a little bit of immunology and then show you some redox chemistry and biology and how it fits into the regulation of immunology as we see it. And I'm going to do that by kind of mixing it in a little bit with some of our ongoing work. So we'll show you some of the most up-to-date stuff that we can think of, at least that I think of. So as was mentioned, I'm a member of the Cancer and Inflammation Program. So we're obligated then by law to show a slide like this, you know, which is the characteristics that lead to malignancy. And this sort of model has been updated, of course, over the years. And those of us that are interested in tumor inflammation are pleased now to know that this has been added. Tumor-promoting inflammation has been added. So in our program, we're very interested in the regulation of inflammation and how that regulation of inflammation can promote or exacerbate disease, and how that regulation of inflammation then can also be played into or taken advantage of in a therapeutic setting for disease. And it's kind of convenient for me that right across the drawing here is deregulation of cellular energetics, because we're going to talk a good bit about metabolism today and redox biology as it relates to the regulation of metabolism. So it's kind of nice that they're right next to one another. So again, by just way of giving you an idea where I'm coming from and where my thinking comes from. This is kind of the overly simplistic, I would argue, model that we think of when we think of cancer development and progression. So you'll have an initial initiation event here, shown by this great little lightning bolt hitting a cell. And then there's an inflammatory response, generally, as the immune system tries to eliminate that target. And this is known as immunoediting, or has been termed immunoediting. And the idea here is that the immune system may recognize this cell because it's damaged and try to remove it. But at the same time, there's often inflammation associated with this event. And if you think of, for those of you that are involved in the laboratories, you know, and you think of the canonical two-hit hypothesis, where a mutation is made and then there is a promoting agent. So for example, in skin carcinogenesis, you add a carcinogen and then you add forvalester, which is literally just an irritant, and irritates the skin, and it drives a lot of inflammation. And this is necessary for the progression of disease. So I've drawn that in a little bit. Some of these cells will escape, is the hypothesis. They won't be eliminated by the immune system. And of course, they'll develop into larger disease. So in my laboratory and in the Cancer and Inflammation Program, we're very interested in how the immune cells are interacting in this mass and how they're propagating the growth of this tumor and propagating the ability of the tumor to escape ultimately into metastases and these sorts of processes. So that's where I'm coming from. So here's a quick kind of outline of what I'm going to try to get through with you here this afternoon. We'll talk a little bit about a very traditional view of redox in immunology. And I'll show you what that means. It's generally based on bactericidal activity, frankly. This is what immunologists think of. And then I'm going to show you that there's a lot more, I think, in redox in immunology. And as I mentioned, it's going to be centered on the ability of redox to regulate metabolic processes within the cells. So I'm going to overview metabolism a little bit. You can tell I'm an old guy. I haven't learned biochemistry in a long time. You guys have probably learned biochemistry a lot more recently, but just in case, we're going to overview a little bit so that we all know we're talking about the right thing. And then we'll talk a little bit about the model system that we use, which are macrophages, a critical pro-inflammatory cell. And we're going to talk about how it's regulated and its metabolism is regulated in part by redox biology. This kind of invariably ends up with NOS. So spoiler alert, NOS is involved in this, as are other redox molecules. So I'll tell you a little bit about how we figured that out, and we'll talk about some of the chemistry. Hopefully I'll be able to tie this back to the things that David told you about a little bit as we go in. And then we'll have a quick review at the end, and then hopefully we'll get out on time. Okay, so immunology in one slide or less, is right here. So for those of you that have had some immunology, you'll recognize that this is painfully oversimplistic. But what I wanted to tell you here is really looking at this from a 30,000-foot view, right? So just to make sure we're all on the same page. Generally, phagocytes at mucosal sites or sites of injury are going to sense a problem. They're going to sense a pathogen, sense a danger. And this results in macrophage activation. And these cells then will elicit or produce a number of inflammatory cytokines. And these are going to coordinate the inflammatory response. And they're not only going to coordinate that primary acute inflammatory response, but they're also important in making sure that the subsequent adaptive response goes as planned. So for example, they'll produce multiple cytokines, including TNF, IL-1, IL-6, and critically a cytokine known as IL-12. IL-12 is fun because what it does is it helps to promote the development of T cells out of what we call a T helper 0 or a starting state into a T helper 1 or Th1 state. And this is driven very strongly by IL-12. And it's these Th1 type T cells that produce a key cytokine in these inflammatory responses that's known as interferon gamma. Interferon gamma feeds back on these same macrophage populations that were infected or sensed the injury in the first place. And you can see that this author has written in very nicely reactive oxygen intermediates and reactive nitrogen intermediates. So it's going to promote the production of redox biology in these cells, which is going to be involved in the elimination of the bug out of these cells. And ultimately, those antigens or bug products that are available are recognized by the adaptive T cell response, and then they can kill newly infected cells, and the system amplifies. So does that make sense as a one slide for immunology? And I think I touched on each of the key things that I wanted to mention there. So I would follow up by saying that, again, from a very naive sense, immunologists, if you go and you say, oh, what do you know about redox in immunology? What they'll say is, oh, right, I think that's how macrophages kill stuff. And that's the way we think, and many of us. And I'll show you that that is the case, of course, but hopefully I'll convince you that there's a lot more to it than that. Okay, so when the macrophages are going to kill something, they have to eat it. Remember, the name macrophage stands for big eater. That's literally what it means. It's a big cell, and it eats big stuff, and then it kills it. And when we zero in on this mechanism of killing is when you see most of the redox biology, okay? So, for example, this is a drawing here of a phagosome that I stole off the internet. You can get the originals, and I recommend that you follow the URLs that I have in here. We'll give you some more information. But here's this big old phagocyte, and it's eaten this bug conveniently drawn in pink here. And what I want to do is just kind of touch on a couple of the ways that the macrophage is going to eliminate this pathogen. One of the first ones are vacuolar ATPases. So these are pumps. They're going to pump protons into the vacuole, lower the pH. And the low pH is important for a couple of things. One is that most cells don't like acidic environments. And so in and of itself, low pH is antimicrobial, but it also facilitates the chemistry of the other redox pathways that are going on in the phagosome. So for example, one of the key ones here, as I've listed, is the NADPH oxidase. And I think this has been mentioned by multiple speakers here in the series, so I hope I'm not being too redundant. But one of the points I wanted to make here is the NADPH oxidase is literally assembled on the membrane of the phagosome. So it's built there. And it's built there intentionally because it is going to shoot reactive oxygen species into the lumen. The whole idea is to put the bugs into a room and dump a whole lot of bad stuff on top of them. So that's what it's going to do. It's going to dump in reactive oxygen species. It's assembled from multiple components, and this stimulation is what causes their assembly into this membrane complex. And as I mentioned here, it's going to direct the ROS into the phagosome. As the name would suggest, it's fueled by NADPH. So an immediate connection to metabolism right away, right? You know, the NADPH has to come from somewhere. So this has to be generated out of the metabolic process of the cell to continue to fuel this process. And I'll show you a little bit about that. Mutations in the NADPH oxidase cause an immune deficiency whereby patients cannot kill cells. They can't kill bacteria that are in the phagosome, and they get a chronic granulomas. They get chronic granulomas. Okay, so what happens here then? A little bit of the chemistry. So this is an iron-based enzyme. It's driven by NADPH, and it produces superoxide. Once the superoxide is produced, it has a couple of routes that it can go through in order to be turned into the good stuff that's going to kill this bug. One of the first ones is that in this low pH, the superoxide will be converted into hydrogen peroxide. Hydrogen peroxide, in turn, is reacted on by the myeloperoxidase, and we'll talk about that a little bit in just a second. This is Fenton chemistry that I know David introduced to you. So hydrogen peroxide, by reacting with iron in the phagosome, produces hydroxyl ions, which are very good at attacking bacteria. So these are the steps then that are going to convert that superoxide through superoxide dismutase, which isn't really drawn here, into hydrogen peroxide via iron into the hydroxyl to kill the bugs, and at the same time MPO, which is a neutrophil protein, will build the hypochlorite, literally kind of building bleach. So it's going to make bleach. And we all know that bleach is good at killing bacteria. So that's what's going on with the NADPH oxidase. So there's a second source of ROS derived from the mitochondria. So you can get a lot of reactive oxygen species out of the NADPH oxidase, but you can also get it out of the mitochondria. So ROS derived from the mitochondria is one that doesn't quite get as much attention as maybe it should. But the mitochondria actually are a tremendous source of reactive oxygen species. Of course, you know, this is a huge redox system as electrons are pumped across the electron transport chain, and in doing so, they force hydrogen atoms out into the intermembrane space, and those hydrogens returning are going to drive the ATP synthase. But it's a less than perfect system. So what's going to happen is that some of these electrons are essentially going to leak out of these complexes. Complex 3 is a big leaker of electrons. It'll generate superoxide. Again, these will be converted by SOD in the mitochondria to produce hydrogen peroxide and some of the same chemistry I just showed you. The other thing I wanted to point out here is that a second direct connection. Now, of course, the electron transport chain is connected to metabolism. I think we probably all know that, right? And it's connected in multiple ways. The first is by NADH, which is produced in the electron, or produced in the TCA and through metabolic pathways. The second is FADH, which is a result of complex 2 in the conversion of succinate into fumarate by succinate dehydrogenase. And we'll talk a little bit more about that as well. So we can fuel this process from two different directions. complex 2 or complex 1. Regardless of which way that fuel comes in, there's the opportunity for leakage at complex 3. Okay. The other thing I wanted to tell you about here is that, that ROS out of the mitochondria can be derived from complex 1. This is a little bit more unusual, but a phenomenon known as reverse electron transport can occur, where electrons actually enter complex 2 and run backwards through complex 1, and the ROS is produced from complex 1. And there's stories coming out in immunology now suggesting that that is perhaps critical in the inflammatory response. Right, so I'm now seeing that this is actually the wrong version of this, isn't it? So hold on one second. Let me just check here real quickly. I apologize for that. I had multiple copies. Yeah, this is a different version. Hold on, can we take a 10-second break and let me swap out the version here? Sorry. Apparently I was not creative in my file names. So I assume that at some point one of you was going to notice, if you haven't already, that it didn't match the handout. Is that right? Let me see. Well, it should be the right version. All right, well, I may have missed a couple of my changes then. Curses, okay. All right, well, I apologize for that. I had too many versions on here, and I think I got the wrong one, but we'll make do. Okay. Sorry about that, but we'll make do. There'll just be a little bit problematic for me, but we'll see how we get by. So, so we have the NADPH oxidase, and we have mitochondrial-derived ROS, right? So one of the fun things that has come out in the literature very recently, just to make sure you're kind of on the cutting edge, is that these are very tightly connected. So in fact, there was a wonderful story that came out very recently that suggested that ROS that's produced by the NADPH oxidase is actually directly involved in eliciting mitochondrial ROS. So these two systems are talking to one another, and if you start to think about it, it really makes perfect sense that the geared up mitochondria will cooperate with the NADPH oxidase to push the ROS production, really get everything into the phagosome and make sure that we can kill the cells. And this was a mechanism that involved the tyrosine kinase figure, and I don't know how much you guys have heard about tyrosine kinases in everything in the class so far, but what it involves is the reconfiguration of the supercomplexes. So you're probably familiar to some extent with the individual complexes, the 1 to 5 complexes that I mentioned of the electron transport chain. Well, these complexes are actually configured into supercomplexes where, for example, you'll go 1, 3, 4, or 2, 3, 4, or different stoichiometries of these protein complexes together. And in a fascinating story, what these authors were able to show was that the NADPH oxidase would activate FIGR, and FIGR would tyrosine phosphorylate, a key protein in complex 2, and it would drive a reorganization of the supercomplexes to favor the activity of complex 2. This allowed a lot of succinate that's fueling complex 2 to come into the electron transport chain and blow out a lot of ROS into the mitochondria. So it was really a very cool pathway and a neat mechanism by which these redox systems are working in collaboration to get the job done. So one of the other reactive oxygen species, or reactive nitrogen species, really, generators in the phagosome is NO. So NOS goes also to the membrane of the phagosome, and I know you've heard a lot about NOS because Dave is very involved, and so lots of the people he has speaking to you like NOS, and you're going to hear even more about it at 5 o'clock, I think, right? So the synthetase for NOS converts arginine into citrulline, is helped again by NADPH, showing you a direct metabolic connection. It uses molecular oxygen, and it generates NO. And that NO then, through chemistry that I won't go into, produces N2O3, which is the effector molecule that will then attack thiols or cause other redox-like signaling. And that chemistry is shown down here, where they're showing a variation on it, which is the reaction of NO with the superoxide that may already be in the phagosome, resulting in peroxynitrite. And this is an area of contention as to how long peroxynitrite is around and whether peroxynitrite is really the effector molecule or whether N2O3 is the effector molecule. But you can talk to Devashree about that, or David, and they'll get you straightened out. So another reactive oxygen species involved from the immunologist standpoint in really killing that bacterium, and that's the full amount. Okay, so let's look a little bit at metabolism. You know, and some people think about this sort of thing when they think about metabolism, and it is horribly complicated, but it's not this complicated, I don't think. And the aspects of it that I'm going to show you today are what I hope I'll be able to tie in, even though I've got the wrong slides for you, but hopefully be able to tie in nicely for you to what we've been doing and the connections in redox. I've put this in here simply so that your handout has one full page. But now, you know, I printed it small in there, so I'm not sure you'll be able to read it. But what we're going to do is we're going to zoom in on this overall view, and I'm going to outline just, first of all, refresh your memory on a couple of these in case you don't remember your biochemistry. And we'll zoom in on a couple of them in some of the key steps, and then we're going to go through and show you how these are tied into to an inflammatory process. So the first one is glycolysis. Most people have heard a lot about glycolysis. It's a big thing in immune cell metabolism now. Everybody says, oh, cells become glycolytic. You know, most of what they're saying on a biochemical sense is incorrect. Glycolysis ends at pyruvate, right? It doesn't end at lactate. Lactate is what you detect when cells make their media acidic, and everybody says, ah, they're glycolytic. Well, they probably are, but glycolysis really is the process prior to that. So it's how you can separate the wheat from the chaff when people start talking about metabolism, right? So a couple of the characteristics that I want you to keep in mind. Glycolysis is, of course, glucose fueled. It's very fast. You know, you've maybe heard of people saying, oh, it switches over to glycolysis. And if you're old like me, you know, you were taught, oh, glycolysis is a crappy way to make ATP. You only make a little bit of ATP. You make all the ATP through oxphos. Well, oxphos is slow. Glycolysis is very fast. So cells can run a lot of glycolysis, and they can rapidly generate ATP. They can also keep their redox in check. So glycolysis is NADH neutral, and only if this is a very clean system. So the system generates NADH at the top, and it burns NADH at the bottom. So it's redox neutral. And if you start to mess with that redox system up in glycolysis, you have to put a whole nother system together to shuttle NADH into the mitochondria. So a variety of changes in the glycolytic scheme will interfere with the NADPH or with the NADH ratios and necessitate other pathways. And I think we'll talk maybe just a little bit about those. It has a net of about 4 ATP. It bifurcates at glucose 6-phosphate, right? Just to remind you that you start with 6 carbons, you split into a double of 3, and you run the bottom part twice for 3. And it has a valve at the end. So it has a rate-limiting step, which is PKM. And I throw that in a little bit as a plug because we work on PKM and think it's a fascinating enzyme. It stands for protein kinase muscle, but it's not a kinase. It's actually a phosphatase. And it generates ATP by stripping a phosphate off of phosphoenolpyruvate to generate pyruvate. And this is a rate-limiting step in the production of pyruvate. And now the cell is going to make a decision. Do I ferment that pyruvate through lactic acid dehydrogenase into lactate and turn the media yellow when I spit it out? Or do I pump that pyruvate into the mitochondria and use those carbons for oxidative phosphorylation? So hopefully at this point you're thinking, oh yeah, some of that sounds familiar from biochemistry. Okay, so the other pathway we'll talk a little bit about is the pentose phosphate pathway. The pentose phosphate pathway starts with glucose 6-phosphate, so it comes off the top of glycolysis, and we think of it as a spillover valve where carbon flows out of the glycolytic pathway. Its big player in physiology, and I would argue in immunology, is NADPH. So this is a rapid, efficient way for the cells to generate NADPH, the stuff that's going to fuel that oxidase. So this is a big source of NADPH for the oxidase. And an experimental validation of this sort of thing is that if you look at the oxidative burst, the ability of a phagocyte to generate a lot of reactive oxygen species, this is almost entirely, in some cells, entirely dependent on sugar because they rapidly bring the sugar in, they shunt it out, and they generate a bunch of NADPH, which is consumed by the oxidase while they produce the oxidative burst. But it's also very important in building blocks. And so the pentose phosphate pathway is a pathway you should think about for cells that have to make other stuff. They have to make other cells. You know, when you're an active cell, you tend, you know, when you think about metabolism, you think about ATP, right? This is the most important. No, what's really more important is making other cells. And the pentose phosphate pathway, among other things, generates the ribose rings that are involved in purine and pyrimidine synthesis, as well as tryptophan, histidine, and then NAD itself. So that carbon gets dumped off to allow the cells to build other cells. You can't build RNA or DNA without purines or pyrimidines. Okay? So that's how I'd like you to think about the pentose phosphate pathway, and we'll touch on it a little bit more. So the last one in our little review here is the TCA and the citrate pathway. So here's the one that you're probably familiar with. Here's the TCA, right? The trichloric acid cycle. It's running clockwise for oxidation. Counterclockwise is redox chemistry in general. Here's the pyruvate that just came out of glycolysis, and it has a destination mainly of running into the cycle where it can be built into citrate, and in some cases that citrate is exported from the cell and involved in lots of other important things. One of the things we'll talk about for macrophages just a little bit is a compound called itaconate, which is really quite a cool story in that when you stimulate a macrophage, it generates a lot of itaconic acid, and it turns out that that itaconate is directly microbicidal. So it poisons a specific pathway in many bacteria, which ironically are pathways that are upregulated when the bacteria are phagocytosed. And I just think it's a really cool example of evolution, you know, kind of playing tennis. So the bacteria figure, oh, I have to exist in this phagosome, so they change their physiology. One of the pathways they turn up is the specific target of itaconic acid. So the macrophage responds by saying, right, if you're going to do that, I'm going to make itaconate. I'm going to kill you anyway. So it includes complex 2 of the electron transport chain. This is another thing that often people don't think about. They tend to think of the electron transport chain as being independent, you know, and that the TCA makes fuel that will go into the electron transport chain, and that's true, but they're actually completely integrated in that complex 2, which is succinate dehydrogenase, is involved in the conversion of succinate to fumarate that permits the cycle to run. It's fueled by pyruvate, which can enter by two different routes. The first route is, as I showed you, pyruvate down for the generation of citrate. And there's a second route that I haven't drawn in here where pyruvate would come down and immediately turn into oxaloacetate. And I'll show you some of the significance of that in a little bit. It can be fueled also by glutamine. So glutamine comes in and enters by anapleresis into alpha-ketoglutarate. And alpha-ketoglutarate then can cycle through. And we're going to talk a little bit about two key components in here. One is alpha-ketoglutarate, and the other is succinate. And I think you've heard a fair amount about them, perhaps in some of the other pathways. And I'm going to hit on those again to make sure that, that you fully get that connection. Okay, the last one is the NOS one. So this is NOS urea and polyamines. And this is where the action is going for arginine metabolism. So arginine is converted into citrulline by NOS2, the inducible NOS that you've heard about from multiple people. It generates NO. It consumes NADPH. So again, a fuel that's built by metabolism is critical for the production of this reactive oxygen species that's then involved in the bactericidal activity that I showed you in the phagosome, connecting those metabolic pathways through redox chemistry. Arginine can be limiting, so the cells possess a nice little mechanism where they can combine aspartate with the citrulline and regenerate the arginine. And this keeps the arginine coming, which can keep the citrulline being made and the NO being made. The alternative is to tap off the arginine into ornithine and then out into the polyamines, including putrescine, which, as the name would suggest, is one of the stinky ones. And in fact, we have a mouse model where we're now looking at this in detail because one of our animal technicians said, oh, the mice are all fine, but you know, when you open them up, they stink. And it turns out that there is a lot of production of putrescine in that system. So it has multiple redox steps. It uses NADPH by NOS. It can produce hydrogen peroxide. So I almost forgot to mention this. So this is another big redox step as you build polyamines. So these are literally kind of a 1-mer, a 2-mer, and a 3-mer of the polyamine units as you build them. If the cell wants to use those, it will use those for proliferation, among other things. If it doesn't, those can be oxidized out by the polyamine oxidases, and they can be pushed back down toward putrescine. And in that process, they generate hydrogen peroxide. So there's a redox cost for that. If you don't use the polyamines, there's a redox cost. If you make the polyamines and you spit them out, then you avoid the redox cost. And in fact, there's some evidence that you might be able to spit those onto somebody else, and then they have to deal with them, and that would be, they would pay the redox cost. So there's, in some models, polyamines are detrimental. So it couples to the TCA by fumarate. So you'll see the common name fumarate that I showed you is also in the TCA. So while we're regenerating arginine, we can then promote over and regenerate or run the TCA by using fumarate. And it's counter-regulated. There's counter-regulation between arginase and NOS, and I think you'll hear more about that from some of the other speakers, but they'll fight over arginine. There's only so much arginine. So if you use it for arginase, you can't use it for NO. So you can regulate NO by the expression of arginine. And as I mentioned, there's a mechanism for replenishing the arginine by aspartate. Okay, so that's a quick little overview. So we're going to move very quickly now into a little bit of a story that I'll try to show you, and I'll point out some other chemistry as we go into this and its connections. So as I mentioned throughout, we're really looking at a lot of myeloid cell biology, and that's because these cells are very plastic. So from a metabolic and redox standpoint, you can push them around, and it makes a very fun system to look at. They also have some very specific activities that allow certain subtypes of macrophages to function a specific way. And then they have specific metabolic pathways such as NADPH and itaconic acid, as I mentioned, that are involved in cytolytic activity. This is, I think, by the way, one of the slides I changed, so it's probably not in your handout. There are suppressive myeloid cells that are involved with tumors, and so that makes it really of interest to guys like us in the Cancer Institute. And they can crosstalk with tumors metabolically. It does involve some redox, but we're not going to talk about that too much today. Lastly, there's immunosuppression. Maybe this isn't lastly, but there's immunosuppression associated with myeloid cells in the tumor. And interestingly, those are redox or redox-related proteins. So NOS2, which I showed you, generates redox, arginase, which controls that, and glucose metabolism. So metabolic pathways that can affect redox pathways are involved in the suppression of immunity by myeloid cells. Of course, they're druggable. That's always fun. That's a good thing when you're trying to help people, is to work out pathways that are druggable. And then you can double hit. So some of these pathways are in the tumor cells, some of them are in immune cells. So if you can find a way to manipulate immunity into a good thing while you're manipulating the tumor into a bad thing, that's the perfect scenario. And so a lot of people have been looking for that sort of activity. And lastly, they even do it, on the Big Bang Theory. So if the Big Bang Theory is doing metabolism, then you should be too, is the way we look at it, right? So, so very quickly then, in what's happening in these metabolic switches within the immune system. So the traditional view of metabolism is that in the presence of oxygen, as we just reviewed, glucose is converted into pyruvate in glycolysis. That enters the TCA and is oxidized, producing CO2 and lots of ATP. The alternative is in the absence of oxygen. Again, the glycolysis makes pyruvate, but now it's fermented into lactate, and you make a lot less ATP. But this guy years ago that I'm sure you've heard about, Otto Warburg, showed that really in proliferative tissues, cells will run this pathway, even in the presence of oxygen. And remember, at first, that was a bit of a conundrum, because we make all this ATP through OXPHOS. Why would we do that? Well, the reason we do that is because you can make it very fast, and you generate a lot of lactate. Lactate can be spit out, and people have now been very interested in why tumor cells do this, but other proliferative tissues do it as well. Otto liked it so much, he said, hey, this is not only something associated with cancer, this is actually the prime cause of cancer. So he was a little bit off on that, but it looks like it's pretty important. Otto also worked on leukocytes. So he studied the metabolism of leukocytes back in 1958, actually, and showed that leukocytes also make this switch. So let's look quickly at some of that. And like I said, what I'm going to do is point out the things that we found in redox in here. So we said, okay, here's these macrophages. We're going to switch them. Now we're going to stimulate them. We're going to take them out of the resting state, put them in a worked up state. And what they do, as I mentioned before, is they move toward glycolysis. And you can see this by assaying the acidification rate. So remember, when lactate leaves the cell, the lactate transporters transport lactate plus a proton. So what happens is, as the lactate goes out, the media gets acidified. And this is what we can read very sensitively in a spiffy little instrument like the seahorse. And what you'll find is that resting cells, have a rate of lactate production. And if you stimulate them with endotoxin so that they think they're seeing a bacterial infection, they become glycolytic. They now produce a lot of more lactate. They're upregulating their glycolysis. And if you graph it, it looks like this. So we look at it this way, where we have cells that are kind of in this resting state. They're largely percolating on, making a little ATP by oxidative phosphorylation. Then we stimulate them, they go to glycolysis, and they become what's unfortunately termed M1. We don't really like those terms, but this is so-called glycolytic commitment. And we call it commitment because while they turn up their glycolysis, they actually turn off their oxidative phosphorylation. So here's their oxidative phosphorylation. And if you poison the mitochondria, you get a drop, so you can see how much oxygen is really consumed by the electron transport chain. Then you can decouple the mitochondria and say, well, look, if I really let them run hard so they're not pushing against a proton gradient, how much oxygen will they consume? That's their maximum rate. And then we can poison it all the way to get to a baseline. So a control cell shows this pattern. An LPS-stimulated cell is pretty flat. So it's turned off all of its mitochondrially derived oxidative phosphorylation. If you graph it, it looks like this. And now this little switch makes a little bit more sense to you, I hope. So they've switched from an oxidative situation to a glycolytic situation. And this was termed by one of the workers in the field as glycolytic commitment, because you can't use oxphos anymore. You are committed to glycolysis. You're committed to sugar. And what does it look like then when you look at these horrific pathways that I just showed you? There's a couple of key substrates that I want you to think about. This is unbiased metabolomics, so we're just looking at the amount of an individual metabolite. And you can see the increase in glycolysis, right? Red shows we've set the wild type resting here to the white. Red shows that the metabolite is going up. Blue says it's going down. So there's an increase in all these proximal glycolytic substrates, and there's an increase in lactic acid. That makes sense, right? We're upregulating glycolysis, collecting a little lactate, spitting the rest out. When we look over at citrate and the TCA, we see that the citrate level goes up. Aconic acid, which is an intermediate, we'll tell you more about here in a little bit, goes up. Itaconate, that one that's microbiocidal, goes way up, which is good, right? These bacteria, these Macs think they saw bacteria. They're making a bunch of itaconic acid to try to kill it. One of the most interesting characteristics, though, that I want to point out to you is this little duplet right here. So alpha-ketoglutarate goes down, and succinate goes up. And we're going to come back to that in just a minute. And this had been, actually, we'll come back to it now. This has been termed a break. As carbon runs through the TCA, it seems to break. There seems to be a drop in alpha-ketoglutarate. I'll show you a little bit more about why that's a break in a second. Now, we know that these cells make NO, and they make INOS, and you can see that down here. A resting cell has a dramatic increase in citrulline. That's coming from the arginine that's being converted into citrulline during the production of NO, whereas ornithine is being produced by arginase. So both enzymes come on, and it makes some arginase, and it makes some NO. Some of that ornithine gets converted off to polyamines, and the putrescine levels go up. So those pathways I just showed you, you can kind of see working as we stimulate the cells, right? So this is what it looks like overlaid. With us, there's an increase in citrate, which allows this stuff to come out and become substrates for enzymes in the cytosol. Not going to go into that a lot, but it also funnels in to itaconate. So this is the itaconate level. And itaconate is really interesting because it is an inhibitor of complex 2. So this plays a part in that oxidative phosphorylation going off, we think, right? So itaconate, which is generated out of the citrate to isocitrate conversion, would come on and can suppress complex 2. I want you to pay attention to this little ratio, succinate up, alpha-ketoglutarate down, because that's going to be important to us in a minute, and I just wanted you to see it in the context of the cycle that I showed you. So why is it so important that there's this weird change in succinate and alpha-ketoglutarate? And the reason it's important is that succinate and alpha-ketoglutarate are cofactors for a variety of enzymes within the cell. One of the most important ones are the PDHs, which I know Dave Wink talked to you about. These are the prolyl hydroxylases. So the prolyl hydroxylases use oxoglutarate, which is alpha-ketoglutarate, and they use that in their enzymatic activity with molecular oxygen to produce hydroxylated proline on proteins, and the byproduct is succinate. So because this is a biochemical reaction, what you can imagine is that if the succinate levels go way high and the alpha-ketoglutarate levels go way down, what will happen to this reaction? It'll slow or stop. And that means that the ability of the PhDs to prolyl hydroxylate goes down. The other thing that I'm sure Dave told you about the prolyl hydroxylases is they are controlled by redox. They are an iron-based enzyme system, and they're controlled by redox. So their reaction, their redox state is critical. So ROS production can shut them down. Succinate and fumarate, as I mentioned succinate, fumarate, just another inhibitor, can shut them down. Hypoxia can shut them down because they use molecular oxygen. And that's really the way you generally think about them, is that they sense a loss of oxygen. But they can be regulated by redox and by these substrates as well. So what happens when they're regulated, and I think this was touched on by a couple of people, including Terry, I think, when he talked to you. So what happens is they stabilize the transcription factor HIF. Does this sound familiar? I hope so. The HIF is acted on by the prolyl hydroxylases. Again, they're converting alpha-ketoglutarate to succinate in the presence of iron and molecular oxygen. And they put this hydroxyl group on. This recruits an E3 ubiquitin ligase, adds a bunch of ubiquitin, and destroys the HIF. So you can have HIF made continuously, but you don't see it in the cell because it's turned over very quickly. What happens when these ratios would change? Now the succinate is very high. Maybe this reaction doesn't run well. And this is a process known as pseudo-hypoxia. So pseudo-hypoxia is the PhDs going off even though molecular oxygen is available. So in this kind of mechanism, we're thinking that the PhD goes off because of that alpha-ketoglutarate succinate imbalance, even though molecular oxygen may be there. Fumarate and succinate, of course, can inhibit them. Once this is off, now the HIF can accumulate and can go into the nucleus, and it can drive transcription. So is that what's happening? Well, we think it is, right? Because remember, I showed you all those red substrates that are glycolytic when the cells are stimulated. Well, it turns out that all of these enzymes, this is just another view of the pathway, everything that's in pink is a target of HIF. It's a target of the regulation by the prolyl hydroxylases. So the prolyl hydroxylases are apparently controlling this glycolytic process and maintaining high levels of these enzymes so that the sugar can begin to continue to flow in and or be tapped off to pentose phosphate. One of the particularly interesting ones is this little kinase down here that we'll talk a little bit more about, because what that kinase does is phosphorylate pyruvate dehydrogenase, the first step in the production of citrate. Okay, so can we see this process actually work? And it's kind of fun because you can. So what you can do is you can take labeled glucose and you can dump it into the cells and you can watch it flow through. You can track the carbons through, okay? So what you can do is say, all right, okay, if I add glucose that's all heavy, the pyruvate will be all heavy. It'll make acetyl-CoA, which is 2 carbons. I'm sure you all remember that, right? And those 2 carbons will combine with oxaloacetate, which is 4, and make a 6-carbon citrate, but only 2 will be labeled from the pulse. So with the mass spec, we can ask, is the citrate 2 carbons or 3 carbons or 4 heavier, because it has C13, than it should be? And that allows us to watch the flux of carbon. And so what you would expect is exactly what you see. So initially, there is some +2 citrate. So the sugar is being made into citrate, and it's too heavy. When you stimulate, look, goes way off, right? Well, it goes off because PDK comes on. So PDK comes on, shuts off this valve. So the pyruvate pathway is now shut off, and there it is. You see it going off. So if you look around the rest of the cycle, you can only see the +2. You don't see any of the +3 because that, you don't see any of the +2 after interferon because the carbon is not making it around. It's been shut off at the source. I told you that those ratios change too, right? So we can look at those ratios and see what had been termed the break. And here it is. If I look at the ratio of citrate to alpha-ketoglutarate, so if I look between here and here, I can see that there's 14 times more labeled citrate than there is alpha-ketoglutarate. So there's a break in here. There's not good flow of that carbon through to alpha-ketoglutarate. And we think that's why the level of alpha-ketoglutarate falls to start to set up that differential that will turn off the prolyl hydroxylases. So the cell says, wait a minute, if I can't do that, maybe I have another route. So it upregulates an enzyme called pyruvate carboxylase that bypasses pyruvate dehydrogenase and enters another way. It sticks a carbon on the end of pyruvate. So this is now 4 labeled carbons. So by looking at the +3 citrate, 3 labeled carbon, by looking at the citrate that's +3, we can see that the switch has occurred. So the cell changes the route of carbon going in, and you can follow that then all the way around the cycle, suggesting the carbon is now entering a different way. It's going out to aspartate, or it's running around through the cycle. And it's driven by pyruvate carboxylase. And other people had seen the same thing, and they said, well, you know, this is because of a breakdown in IDH, and that's why you get this change in ratio, but we think that's wrong, and I'll show you why in just a second. So some quick primary data that I'll just go through very quickly because it's not the big goal of what we want to do here, but what we find is that this cytokine, this regulatory cytokine IL-10, is dependent on that switch to glycolysis. So you get IL-10, if you interfere with the use of sugar with 2-DG, you get less. If you stimulate them in limiting sugar, you get less IL-10. We can map out the mechanism and show that it's ATP synthesis that falls. The phospho-AMP kinase comes on, suppresses TORC, and prevents IL-10, which you can recapitulate by suppressing it directly. So we built ourselves a little model, because it's always fun to build models, that when you stimulate, you make 10, it regulates back, and that should regulate the glycolytic commitment. And in fact, it does. So if you look at an IL-10 null cell, they overshoot. They go so far to glycolysis, they're almost going off the end of the cliff. But what's fun from a redox standpoint is I want to show you how that works, and that works through a redox molecule. It works through NO. So what we notice is when you stimulate cells, and you can look at their oxidative phosphorylation going off, you can stimulate with a variety of different things. Some things work, like this PAM-cis and interferon. Some things don't. And it turns out that that's in direct correlation to their ability to produce NO. So if you can produce NO, you crush the OCAR. If you can't produce NO, you don't crush the OCAR. So is it NO? Looks like it is. You can add it directly and crush the OCAR. So that whole part of commitment, we think, is due to the redox activity of NO. So NO can hit by nitrisation of thiols that Dave described to you, I think. So here it is, SNO. Hitting thiols in complex 1 had been shown as a mechanism. And because NO can compete with oxygen binding sites, it was also thought that it could interfere with complex 4 of the electron transport chain. Both of those would allow the NO to shut it off, right? So let's see. Well, if we add Ditano, the oxphos goes off. That's good. If we make the oxphos go off, but we do it in a NOS null, no effect, right? So it's entirely dependent on the NO. And in fact, we can show that the IL-10 regulates that because IL-10 null cells make more NO. So the cell, and I think I summarized it here, so the cell is regulating glycolytic commitment by the production of IL-10 and its effect on the redox molecule NO. Okay, so then if we knock out NO through iNOS, as I showed you, we get some interesting changes in overall. You can see they can still upregulate glycolysis, but look at the switch here now is gone. Look at the ratio now between succinate and alpha-ketoglutarate. The alpha-ketoglutarate levels don't fall. So there's still a difference here, but it's not as sharp as it is here. We know our experiment works because you see the citrulline doesn't increase. So we know that, you know, thank goodness that we put the right mice in there. So what happens now when you look at the carbon? Okay, if we look at the carbon in the NOS nulls, which are in the warm colors here, what happens is you don't see the M2 go off. You don't see this valve close, and you don't see the M3 come on. So you don't see this pathway come on. And sure enough, if you look at the genes, you'll see here, this is the upregulation of the kinase that shuts it off. Doesn't come on in the NOS null. Here's the upregulation of the enzyme that bypasses. Doesn't come on in the NOS null. Right? This is a HIF target. So it's telling us that the prol hydroxylases are not functioning properly to turn PDK1 on. So it was a big clue to us. So if the prol hydroxylases are not functioning properly, and if they are NO dependent, how is the oxphos going completely off? Okay, these guys had said it was IDH1, but they don't know anything about redox biology, because if you look at IDH1 and you think about redox, and Dave showed you a bunch of different ways, right, that NO, for example, can hit. It can hit through nitrisation, it can hit through peroxynitrate, et cetera, right? But that doesn't work very well on IDH1. There's no indication that it should work. And in fact, IDH1 is a cytosolic protein. So who's writing a paper saying that it controls the TCA in the mitochondria anyway, right? So I don't think these guys really were thinking about redox biology. But we were, and we said, hey, there's this other enzyme up in here called ACO2, and it's involved in that same sort of step, okay? So if NO is required for this little break to occur, right? Then NO, if it's IDH1, then NO should be required for that. Well, IDH1 goes down, but it goes down even when you block NO or even in NO nulls. So it's not IDH1. They got it wrong. They don't know it yet because we're just writing it up, but they're going to find out soon that they're wrong. So, well, I'll even skip that because I don't want to run out of time. I'm already running a little behind. So let's look at the aconitase then. So the aconitase is a much better target, and I think Dave showed you some of the chemistry as to how NO and oxygen radicals and redox biology would hit aconitase. And I'm not going to harp on that too much more than to remind you that citrate to isocitrate is how aconitase works. That's the reaction, and it's a two-step reaction that produces cis-aconitate in the middle. And if you remember, this is the thing that's converted into itaconic acid that makes the antimicrobial. It sucks it out of the middle. Okay? If you look at the active site of aconitase, it's an iron sulfur cube. So it's a cube structure, and it's very subject to redox attack. And actually, there's old stuff in the literature that shows that NO will oxidize one of these irons and kick it out of there and destroy the cube. And when you destroy the cube, the aconitase loses its activity. And in fact, then you can go back, take that aconitase, add in iron and reducing agents, and fix it and put the iron back in. So it was a really good target. And in fact, when we look, we find that the stimulation turns off aconitase activity, and that requires NO. So if we block it with immunoguanidine, it all goes away. So IDH1 is not the target. This is redox biology hitting aconitase. And we can prove it even further by permeabilizing the cells. And this really put it to bed. So here's your TCA for comparison, right? Just an ultra simple one. So when we do the assays of activity, what you do is you permeabilize the cells. You wash all the substrates away, okay? And then I only add in the fuel that I want. So I wash all the substrates away, and I add citrate. And a regular cell, the black line here, can do okay. This is oxygen consumption. So it can take that citrate, and it can run it a couple of steps, generate some NADH, that'll go into complex 1, and I'll get oxygen consumption, okay? So that's this. So, you know, there's a little bit of decline in efficiency, but hey, your efficiency would fall too if I poked holes in you, right? But now, if I look at one of the ones that has been stimulated, there's a plummet. So they can't use citrate, right? Because there's a break here. That's what I showed you. There's this break between citrate and alpha-ketoglutarate. But check it out. If you come in now and give them citrate, now they work fine. So the only enzyme pathway that I'm skipping is aconitase. So if I bypass the aconitase by giving them the product of aconitase, now they work fine. That means that the NO didn't nitrosate complex 1 and turn it off. It didn't mess with those. What it did is it stopped aconitase, and that's how it's turning off the pathway. And so we can look at that very specifically, and of course, in the nulls, it doesn't work. They'll accumulate lipids too, and I probably shouldn't even put that in there, so we're going to skip it. I wanted to come back to here just to remind you that we have that mitochondria-derived ROS is absolutely a big deal here, and can still control the metabolic activity because this ROS starts to hurt the mitochondria. And one of the last things I'll show you here in the actual redox biology is that the inflammatory cytokine IL-1 is also directly tied to these processes, because once we start to mess with mitochondrial function, either directly by NO or by turning off aconitase, ROS production out of the mitochondria leads to oxidation of mitochondrial DNA, and this activates a multi-protein complex called the inflammasome. Immunologists love the inflammasome because what happens is as cells make interleukin-1, and it's true also for interleukin-18, there's a pro-cytokine made. And it is then cleaved by this complex, this inflammasome complex, into mature IL-1, which is secreted. So the secretion of IL-1 and subsequent inflammation is tied directly to mitochondrial ROS, which is tied directly to the redox biology and the control of mitochondrial function. The last thing I would show you, I should probably skip over this because I'm late, but gene regulation is not so affected except for IL-1, as you might anticipate. And again, I apologize for putting the wrong talk on here. So I had some great summary things at the end. I'll leave you with this, which is a little busy, but work with me for just a second, and you'll see there's lots of fun redox in here. So this is kind of an NO-centric view of programming, right? So what I showed you is that this redox molecule directly can reduce oxidative phosphorylation. It can suppress aconitase 2, which is controlling the conversions between citrate and alpha-ketoglutarate so that alpha-ketoglutarate levels fall. This differential is out of whack. The PhDs go off. HIF activity goes up. That's been reported to be involved in the production of IL-1. I didn't want to go there for you, but it drives, as I showed you, the kinase PDK1, which turns off pyruvate dehydrogenase, causes the carbons to shift, and helps to replenish the arginine that promotes NO activity. So it runs in a nice clean circle. At the same time, before this aconitase goes off, that citrate that goes out is sequestered into the production of itaconate, which will be involved in lysis of the bacteria. And the whole thing, we think, is regulated by IL-10. And the last thing is that there's other metabolic controls in immunity, obviously, and you can read about these. Regulation of glucose utilization, regulation by PKM2 is now coming out, as I told you a little bit about. The macrophage one is closer to home for me. That's why I picked on it. It's also more clearly a redox activity. That's why I picked on it. But I would recommend that you grab up one of these reviews, and you'll see a little bit more of the connections there. Just a couple of final thoughts then. So the redox biology touches lots of aspects of immune cells. All of these species, ROS, NO, NADPH, play important roles in metabolism and in the innate immune effector function. The activity should be matched to a specific niche, and we can talk about that a little bit more sometime, but lots of cell biology is done the wrong way, right? I remember going to a talk several years ago. It was a great guy. I was like, why are we studying human cancer in a dish with 21% oxygen, right? Tons of sugar and cow serum. You know, I mean, how far from reality can you get? So when you think of metabolism, you have to think of what's available to the cells, and it's a whole nother way to think about this stuff. Efforts are underway to reprogram macrophages in cancers to make them match those niches or fix those niches, and many of those will induce redox molecules like NO or be regulated by them. I'll also point out that most therapies that work utilize interferon gamma, which is a great inducer of redox species, including NOS, and it's also a good driver of mitochondrial ROS. And finally, the NOS, and you've maybe heard about this from some other speakers already in the course, but NO production is dramatically different between mouse and human. You know, and if you go and you look at how cancer therapies are tested, they're tested in mice. So should we not be surprised that they don't work when we go to human beings? I think what we're finding is that all of these metabolic changes that are based on that redox pathway are going to be different in mice and humans. So we need to do it. I take credit for lots of fun people up in my lab. A lot of the data I showed you was done by Erica. Luke has done a lot of our metabolic work as well. Some of the IL-10 stuff was done by Walt, but we fired him because he drinks too much. And this is the current group up here. So I apologize for going over a little bit, and worst of all, for having the wrong slides so they didn't match for you, but I'm happy to clarify anything I can for you. Thanks.
So what are the levels of NO in the macrophage? Do they fluctuate with the immune response?
Yeah, so it's highly, highly inducible. So in murine macrophages, we'll make micromolar concentrations of NO very quickly. So within 8 or 10 hours, you'll see very high levels. we've actually been trying to look now to say, is this physiologically relevant, right? Are there, in a dish, there's a lot of cells in the dish. We can control their density. We think, and Dave's lab and Debashree could comment on it too, have evidence in vivo, even in human beings, that there's evidence of NO production. So you can see effects of NO. You can see the chemical byproducts in the tissue. So you know that the levels have gotten pretty high. But it comes on extraordinarily fast. Some of the effects are cell density dependent, and we think that that's because, as again, you've heard from Dave already, there's different chemistry for NO given the concentration. So when you put them together in high densities, you get very high levels of NO, and you'll get certain chemistry that you may not get at lower levels, if that makes sense. There's relatively little known about how it goes off, actually. We asked Dave's lab, and they've done some, but we're now looking at how the message goes off and how transient that may be. Now that we know that a contease, for example, will be hit, we find that a contease has to be replaced. That's a modification that the cell won't fix, so it has to build new aconitase. So the mechanism by which it hits and the continuing production is going to be important, I think. So.
Need that microphone.
Yes.
Okay, we have one announcement, that being next week we have our final class. So I'm working on the final examination, and basically it's a multiple guess exam. It'll be posted on the website, and there will be one question from each lecture. And if you get 70%, you pass, and then you get your certificate. So the exam will be posted for about a month and a half. So you'll have plenty of time to take it. And it's open book, open notes. If you have your handouts, you should do just fine. Okay, so our last speaker today is Debashree Basudar, and she got her PhD at the University of Arizona. Subsequently, she was a postdoc at the University of California, San Francisco, and she's been at NCI now for 2 years. And the title of her talk, Chemical Biology of Nitric Oxide and Nitroxyl.
All right. Okay, so I'm a research fellow in Dr. David Banks' lab, and so I'm going to talk about the chemical biology of two small molecules, nitric oxide and nitroxyl. So I'm sure you have heard a lot about nitric oxide, so I will give you a brief background. So nitric oxide was discovered by John Presley in 1972. But initially, the biological effects of nitric oxide was studied in terms of environmental toxicology, because at that time, nitric oxide was known as an air pollutant. Like nitric oxide and many of the nitrogen oxide species were found as air pollutants. Also, nitrogen oxide species occur as a carcinogenic compound known as nitrosamine in stored food. So most of the time, people studied the health effects of those toxic compounds. It was not until 1998 Nobel Prize was given in the field of nitric oxide, which established the role of this molecule in biology. So it was shown in late 1980s that nitric oxide is biosynthesized by an enzyme called nitric oxide synthase. So it's a two-step monooxygenation reaction where L-arginine is first converted into N-hydroxyl L-arginine, which is then converted to L-citrulline with production of NO. So there are three different isoforms, NOS1, NOS2, and NOS3. NOS1 and NOS3 are constitutively present, while NOS2 is the inducible isoform. Other than enzymatic pathway, NO can also be produced in a non-enzymatic way from nitrite. So this process can play a role in under hypoxic condition because, as you can see here, NOS enzyme is oxygen dependent. So under hypoxic condition, nitrite can become the main source of production of nitrite. So these are some of the reactions of nitrite. So the reactions of nitrite, which so these are some of the direct reaction where nitric oxide reacts directly with mainly metal centers or with free radical molecules. So NO has a unpaired electron. So metal reaction with metal and free radical reaction are the most important reactions. Apart from that, nitric oxide can also react with oxygen to produce some of these species, which Dan already talked about. So NO2 can lead to oxidation and nitration reaction, while N2O3 can lead to nitrosation. So in biological system, tyrosine nitration is one of the molecular signature of this step. And then nitrosation, like the two amino acids which undergo nitrosation is thiol or amines. So as for the direct reaction, one of the important targets of nitric oxide is soluble guanylyl cyclase, also known as SGC. So SGC is a heme protein with an iron 2 oxidation state with a histidine residue at the bottom of the heme plane. So when nitric oxide binds to it, the histidine residue comes off, and this causes change in the structure of the enzyme. So this change increases the activity of SGC by like 200 folds. And this enzyme then converts GTP into cyclic GMP, which is a second messenger molecule, which is responsible for many of the properties of nitric oxide, such as many of the cardiovascular function and muscle relaxation and all, like many of the other properties under physiological condition. Nitric oxide role in cardiovascular function has been shown for a long time, but since it's a radical molecule, it can also react with other radicals, leading to quenching of chain reactions, forming RNO species. It also plays an important role in Host-Stephen system under different pathological or microbial attacks. In our lab, we are mainly interested in the cancer biology of nitric oxide, which I'm sure Lisa talked about quite a lot in the last class. So then the other molecule that I'm going to talk about is HNO. So HNO is a reduced form of nitric oxide. However, it has not been convincingly shown to be produced under physiological condition. There are many reports where people say that they have evidence for formation of nitroxyl under physiological condition, but one of the limitations for detection of nitroxyl is it has a really fast self-dimerization rate. So these are some of the reactions which are reported in the literature as evidence for formation of HNO. So one of the key pathway by which HNO can form is from this intermediate of NOS reaction, L-hydroxyl L-arginine, which is also known as NOHA, and this can undergo oxidation to form HNO. Also, it can form through NOS when there is a lack of tetrahydrobiopterin, which is a cofactor for this enzyme. Other than that, hydroxyurea, hydroxylamine can also undergo oxidation reaction to form HNO. But one of the most recent studies have shown that a small SNO molecule, which is HSNO, which is the smallest SNO molecule, can react with H2S, which is another transmitter, to form HNO. So these are some of the common reactions of HNO. While NO reacts with ferrous heme and free radical, HNO mainly reacts with ferric heme and ferrous heme, as well as thiol species. So the reaction of HNO with metal centers is known as reductive nitrosylation, and this is mainly used for detection of HNO. Also, other methods which are used for detection of HNO is this reaction, that is reaction of HNO with thiol. So as I've mentioned earlier, HNO can also undergo self-dimerization to form N2O, which can be used as a molecular signature for detection of HNO. So detection of N2O and HNO has been used by mass spec methods as confirmation of production of HNO. So other than that, HNO can also form, can react with oxygen to form HNO oxygen intermediate. This molecule has not been convincingly characterized. Some people believe that it is similar to peroxynitrite species, but there are some differences between this species and peroxynitrite, because peroxynitrite can only cause single-stranded DNA damage, while this molecule can cause double-stranded DNA damage. So this intermediate can lead to protein nitration and hydroxylation. The biological effects of HNO has been shown in terms of alcoholism, cardiovascular effect, where it is used as a preconditioning agent. And we have shown the effect of HNO in cancer biology and inflammation, which I will show in my later slides. So under physiological condition, these are the most relevant reactions of HNO. As I mentioned, it reacts with ferric heme to form ferrous NO. It can also react with ferrous heme to form meth hemoglobin sort of species. It can react with thiols to form a GSSG in the presence of excess thiol or these N-hydroxysulfonamide species. So these are some of the relevant rate constants, and these the rate constants are not only important in biology, but they have been utilized for development of methods for detection of HNO. So one of the key reactions under biological condition, as I mentioned, was reaction with thiol. So it can react with thiol to, RNS can react with thiol to form RSNO, which can again be converted to produce HNO in the presence of GSH. HNO can also be produced with NO, which can further react with thiol to form N-hydroxysulfonamide. This species under excess thiol can lead to formation of GSSG sort of species, and production of hydroxylamine, which has been shown in our group to produce HNO again. So as I mentioned, one of the limiting factors in the chemical biology of HNO is the detection. So these are some of the methods which are used for detection. So Angeles salt has been used as a HNO donor for a long time. It is a pure donor of HNO, but it also produces nitrite as a side product. So this is a meth myoglobin assay, which is shown here in this red trace. In the presence of HNO, it completely converts to MBNO to form peaks at about 530 and 560 nanometer, while DNO, which is a pure NO donor, does not show these peaks. So it basically stays as meth myoglobin. This reaction was further proved by addition of GSH, which quenches HNO. So in the presence of GSH, we do not see formation of the reductive nitrosylation peaks. The other method which is commonly used is this electrochemical method. So this is again an electrode for nitric oxide detection. So we use indirect method to convert HNO to NO by using ferricyanide. And as you can see, the red bars show the NO formation at different pH in PBS. And as in the presence of ferricyanide at a higher pH, we see that there is HNO formation, which is evident from the blue bars. So the difference between the red bar and blue bar shows the amount of HNO formation. And as you can see here, that as the pH of the media increases, the HNO formation also increases. At a lower pH, there is no HNO formation. So recently, there has been a lot of research going on for in vivo detection of HNO, and these are some of the fluorescent probes that have been generated over the years. So this fluorescent probe was generated in Dr. Lippard's group in MIT. So this can distinguish between HNO without interference from NO and peroxynitrite and hydrogen peroxide species. But one of the disadvantages of this probe is this can be reduced also by thiols. So cysteine will actually show fluorescence with this probe. So there has been a lot of research going on in this field, and this compound, which was recently published, this one does not show reactivity towards thiol and shows much more specificity for HNO. The other class of compound which are used for detection of HNO is these phosphine-based fluorescent probe, which were developed in Dr. Bruce King's group in Wake Forest University. And as you can see, HNO can react with this species, which then reacts at this oxygen site, and the fluorophore is released, which leads to the fluorescence of this molecule. The other class of molecule which are being developed is these Tempol-based fluorescence agent. And on reaction with HNO, it forms this species, which shows a strong fluorescence. So as I mentioned before, HNO chemistry is limited by its self-dimerization rate, forming N2O. So to study the chemical biology of HNO, we need donor molecules. So these are some of the donor molecules for NO, which are commonly used, and these are some of the donor molecules for HNO. So the most common donor molecule for NO, which has been long used, is organic nitrate. And one of the example is nitroglycerin, which is used for angina pain. So this molecule was first discovered by Alfred Nobel, and this was like basically the precursor for explosives. But when workers at that time were working in his factory, he noticed that people would get headaches because of exposure to vapors of these molecules. This was because there was like dilation of their veins in their head, which was leading to this headache. So this has been used for a long time for heart-related angina pain. The other molecule which is used commonly is isosorbate dinitrate. And both these molecules are catalyzed by cytochrome P450, so to release nitric oxide. So the cytochrome P450s that are involved is CYP2E1, CYP2J2, and CYP3A4. The other class of nitrosyl NO-releasing molecule is metal nitrosyl, and these are mostly used for acute hypertensive stress. And these molecules have S-nitrosylthiol-based molecules. This is SNAP, has been used for inhibition of platelet aggregation. So we are mostly interested in diazinium dilate-based molecules because these can release NO as well as HNO under spontaneous condition, and it does not require any enzymatic reaction. But let me go over some of the HNO donors first. So this molecule in jelly salt and this molecule, pilotis acid, was synthesized in the late 1800s. So it was synthesized in like 1896. But there wasn't much of research going on at that time in the field of HNO. So they are still used for studying the chemical biology of HNO. So this molecule under physiological condition gets protonated at this position. Then it undergoes a slow tautomerization reaction to transfer this proton on this nitrogen, which then decomposes to form HNO and nitrite. While pilotis acid shows HNO production only at a higher pH. So this molecule is mostly used as an HNO donor under physiological condition. The other molecule which is used as an HNO donor mainly for treatment of alcoholism is cyanamide. So this is catalyzed by catalase enzyme, which then releases HNO. But one of the side effects of this molecule is the production of HCN. So for treatment of alcoholism, even though this is approved in Japan and European countries, and US, it's not approved. The other HNO donor is this class of molecule, which is known as nitrosyl compound. And recently, there has been compounds, ruthenium-based compounds, which are activated by thiols to produce HNO. So we like to work with diazinium dilates because these can be tuned easily by different substitutions on the backbone to release NO versus HNO. So when the amine group is when the amine is a secondary amine, it's a pure HNO donor, and it releases two equivalents of pure NO donor, and it releases two equivalents of NO. While these molecules, when the amine is a primary amine, it releases one equivalent of HNO and one equivalent of NO. And this ratio actually depends on the pH of the solution. So we can change the pH and make it release completely pure NO versus HNO. This class of compound was extensively used and developed by Dr. Kiefer's group in NCI as a research tool, as well as for different pathophysiological processes. So one of the important things about these molecules is they can be derivatized at this O2 position, and that can lead to site specificity of NO or HNO release. So these are some of the molecules which were developed in Dr. Kiefer's group. And as you can see here, as the amine backbone, secondary amine backbone changes, the half-life changes. So for L-proline, prolino is the diazinium dilate. It has a half-life of only 2 seconds, while detano has a half-life of 20 hours. And when it is protected at this O2 position, the half-life can increase to up to 6 days or more. So the primary amine diazinium dilates can, as I mentioned, can release both NO or HNO. This is because this proton here can undergo tautomerization reaction to shift to this, and then this molecule can decompose to produce HNO. But under a low pH condition, this reaction does not occur, and it basically undergoes this process to release 2 molecules of NO. So as you can see, this reaction cannot occur for secondary amines. So secondary amines can only go through this process and release pure nitric oxide. So this class of compounds show a peak at 250 nanometer, which is commonly used for quantification and for studying their half-life. The reason we basically use this absorbance 250 band for quantification is because these can have nitrite and other impurities. And if you make a solution by just weighing it, you may not get the right concentration. So we were also interested in expanding the library of primary amine-based diazinium dilates so we could get more HNO donating compounds. In this effort, we use these cyclic amine, primary amine cyclic molecules. So we made CPNO, CH, cyclohexylamine-based nonoid, cycloheptylamine, and cyclooctylamine-based nonoid. And as you can see here, the half-life basically changes from like 3.7 minutes to a maximum of 6.4 minutes. So the half-life did not change much. So for the secondary amine-based nonoid, the half-life variation was much more. So then we looked at how much NO and HNO is being released from these molecules, and we found that cyclopropylamine nonoid released the maximum amount of HNO at a physiological pH. Then we studied the cytotoxicity of these molecules, which do not show cytotoxicity until we are in a millimolar range. And as expected, they only showed cytotoxicity in a millimolar range. So at a low concentration, they are not cytotoxic by themselves. So since the half-life didn't change much, we synthesized O2 derivatized molecule by using this ACOM protecting group. So we studied the mechanism of decomposition of this molecule. So in the presence of esterase, this molecule, which has a peak at 240 nanometer, shifts to 250, which is the peak for a diazinium dilate, and then it decomposes with a half-life of 5.8 minutes, which is basically the same for the parent molecule. But in the presence, when there is no serum present, this 240 peak decomposes with a clean isobestic point. So it is not going through the parent molecule. So we further investigated the mechanism of decomposition, and we found that this proton can be deprotonated easily, and then it can form this intramolecular cyclic ring, which can then form these nitrosamine base compound, which decomposes to form HNO. So when we have this O2 protection, and when there is no serum present, these molecules are pure HNO donor. So we quantified the amount of HNO production, and it was like 97%. So in our lab, we are mainly interested in looking into the effect of various HNO and HNO donating molecules in breast cancer. So these are some of the breast cancer statistics, and I would skip through it. I just want to mention that we mainly work on triple negative breast cancer, which are estrogen receptor negative, progesterone receptor negative, and HER2 receptor negative. So triple negative breast cancer accounts for only 15% of breast cancer patients, but these patients have a poor survival rate. So inflammation is a key driver of tumor progression in breast cancer, and NOS2 and COX2 enzyme are two of the key enzymes involved in this process. In cancer, the chronic inflammation can result from like lifestyle, obesity, infection, or autoimmune disease, which can then lead to increased level of RNO, reactive nitrogen species, PG2, which is produced by cyclooxygenase, pro-inflammatory cytokines, or matrix metalloprotein rearrangement. And under normal circumstances, like in the physiological system, we have a very good immune surveillance. So most of the time, these mutations which occur, our immune system is able to take care of it. But under some circumstances, this chronic inflammatory conditions can lead to tumor formation. These are some of the common therapeutic therapies which are used for breast cancers. One of the common therapies is radiation. Then we have hormonal therapy, like use of tamoxifen or aromatase inhibitors. Then chemotherapy is used, and immunotherapy also can be used, even though it is not that popular in breast cancer. But multi-drug resistance is one of the key problems for triple-negative breast cancer patients. So our goal is to develop new therapies for this type, the triple-negative breast cancer patients. As I mentioned, NOS2 is involved in progression of various cancer, and the type of cancer which are shown here in green show poor prognosis related to NOS2 upregulation. And as you can see, breast cancer is one of them. In cancer biology, nitric oxide shows a concentration-dependent role. At a low concentration, it can lead to deamination, which can lead to mutation in the DNA. It can also lead to increased angiogenesis and invasiveness, and all these processes promote tumor progression. But at a higher concentration of nitric oxide, it leads to apoptosis and inhibition of angiogenesis, which can lead to tumor cell death. So in our lab, we use these diazinium dilates, and we have found that depending on the time and flux of nitric oxide, it can lead to activation of different molecular signature. For example, at a low nitric oxide concentration, ERK is phosphorylated, and this level basically can be correlated to the amount produced by NOS3. And as the NOS production increases, HIF1 alpha can be stabilized, and at a higher concentration, P53 is phosphorylated, and then there can be nitrosative stress-mediated signaling, such as modification of thiol residues. So this concentration can be achieved by NOS2. In a cancer cell, the flux of nitric oxide is believed to be around 300 nanomolar, which is in this range. So based on the flux of nitric oxide in the cell, the outcome of the disease can be modified. So from therapeutic point of view, there can be two approaches. So we can either inhibit NOS2, or we can increase the amount of NOS2 so that it leads to apoptosis. So one of the previous postdocs in our lab, in collaboration with Stefan's group, showed in a patient cohort that 73% of breast cancer patients have high NOS2 expression. And when she plotted them based on their ER status, we found that ER negative patients show like poor survival when NOS2 is high. So 92% of deceased ER negative patients had high NOS2. And in a recent patient cohort, which was done in Ireland, it has been found that 100% of the deceased patient had high NOS2. So the other enzyme which plays a key role in inflammation is COX-2. So COX-2, this is the mechanism of action of COX-2. So phospholipids can be converted by phospholipase A2 to arachidonic acid, which is the substrate for COX enzyme, which can lead to formation of different prostaglandins. So COX-2 has been found to be correlated to different types of cancer, which are listed here. And breast cancer, as you can see here, shows correlation with increased COX-2 level. So COX-2, cyclooxygenase enzyme exists in two different isoforms, COX-1 and COX-2. COX-1 is the constitutive isoform, which is mainly responsible for normal physiological processes such as maintenance of stomach lining, platelet aggregation. On the other hand, COX-2 is the inducible isoform, is mostly responsible for various pathophysiological processes. So some of which are inflammation, pain, and cancer. So these are the different publications which have come out showing the role of COX-2 in breast cancer. So from the same patient cohort data, when we looked at the expression of COX-2, we found that the patients which had high COX-2 and high NOS2, they had a hazard ratio of 20. So compared to this is high NOS2 only. So the hazard ratio increases further compared to patients with only high NOS2. So in our lab, we are interested in looking at this group of patients. So we have been studying the effect of this loop, but I am not going to go much into that today, but I'm going to talk about some of the molecules which we have synthesized in our lab. So these are molecules where we conjugated a dizenium dialate moiety to different NSAID, non-steroidal anti-inflammatory drugs. So the non-steroidal anti-inflammatory drugs that we have used is aspirin, which is an irreversible inhibitor of COX-1 and COX-2. we have used indomethacin, which is a reversible inhibitor, and we have also used niflurimic acid, which is a reversible inhibitor of only COX-2. And as for the dizenium dialate, we compared DINO, which is a NO donor, to IPANO, which is the HNO donor. So non-steroidal anti-inflammatory drugs are commonly used to alleviate pain and inflammation. And these are some of the common NSAID molecules. They work by inhibition of cyclooxygenase, which is responsible for conversion of arachidonic acid to different prostaglandins. So one of the main side effects of NSAIDs is since they inhibit both COX-1 and COX-2, this leads to gastric ulceration because COX-1 is responsible for maintenance of stomach lining. Another class of molecules which were known as COX-CEBs, which were specific COX-2 inhibitor, were synthesized. So since these molecules are larger in size, they specifically fit into the active site of COX-2 without inhibiting COX-1. So even though it's inhibited COX-2 specifically, on long-term usage, it led to cardiovascular increase in cardiovascular events. So for this reason, Vioxx and celecoxib, which were taken out of the market, and Merck had to face a huge lawsuit because of these class of molecules. So our approach, we used NO donors and HNO donors because, as I mentioned earlier, NO can increase NO can protect the stomach lining as well. So we thought that these conjugated molecules can protect against the side effects of aspirin. So we wanted to make sure of that. So we studied the effect of these molecules on ulcerogenicity. And as you can see here, this is the control group, and this is the aspirin group. And we basically gave them a high dose of aspirin and the equivalent drug. And then these stomach bleedings were counted after 6 hours. And on treatment with aspirin, there was a huge increase in ulceration index. With dinoaspirin, there was no ulceration. And this was the first time we showed that ipanoaspirin, which releases HNO, can also protect against ulcerogenicity. So both dinoaspirin and ipanoaspirin protects against ulcerogenicity. The other side effect of this class of COX-2 class of molecule was heart-related problem. So we studied the effect of ipanoaspirin on cardiomyocytes, and we found that it increases the contractility of cardiomyocytes. So it basically increases, it basically preconditions the heart against heart attack. So when in the event of a stroke, the damage is much less. We also wanted to make sure that it still retains the anti-inflammatory property. And as you can see here, it still maintains the anti-inflammatory property compared to aspirin. So it shows similar anti-inflammatory property. Then we investigated the effect of these molecules on different breast cancer cell lines. We used MB231, MB468, which is ER negative breast cancer cell line, and we used MCF7, which is ER positive breast cancer cell line, and we used MCF10A, which is a non-tumorigenic cell line. And as you can see here, ipanoaspirin showed the most toxicity in MB231. It also showed the data was similar for MB468 and MCF7. The dinoaspirin molecule, which is NO releasing, showed less cytotoxicity, but it was still pretty toxic. The interesting thing about both these HNO-aspirin and NO-releasing aspirin is it did not show any cytotoxicity on non-tumorigenic cell line. Then we looked into the effect of these molecules on angiogenesis using a Huvex branching assay, and this is the control, and it, as you can see, it shows a branching, and this branching was inhibited by addition of low dosage of dino and ipano aspirin molecules. Then we studied the DNA damage using COMET assay. And so this is our control, DMSO control. This is our positive control, where we induce DNA damage using hydrogen peroxide. Ipanoaspirin showed DNA damage, but NO-releasing dinoaspirin did not show DNA damage in COMET assay. The other thing which we investigated was the ability of these molecules to inhibit COX-2. So we used prostaglandin E2 as the readout of COX-2, and we used the Cayman kit for this assay. And we found that ipanoaspirin inhibited PGE2 levels in a concentration-dependent way, while dinoaspirin did not have much effect. While part of this effect is for sure from the aspirin moiety, we have also looked at the effect of Angeli salt, which is a pure HNO donor, and we have shown that it inhibits, it can inhibit COX-2. We believe the reason for this can be, so there is a ferric heme at the active site of COX-2, which can undergo reductive nitrosylation in the presence of HNO, or it may be a thiol-dependent process. So we haven't investigated the mechanism, but these are the most two common pathways which have been shown for different HNO donating molecules. We also investigated the effect of HNO releasing ipanoaspirin and dinoaspirin on GABDH. And we found that ipanoaspirin inhibited GABDH activity in a concentration-dependent manner, while dinoaspirin did not show any effect. This has been shown due to the inhibition of a critical thiol residue at the active site, which reacts with HNO to form this sulfonamide residue. And this is an irreversible modification. Then we investigated the effect of these molecules on our animal model. So we used News mice and we used MB231 cells labeled with GFP. And as you can see here, this is our control group, which was the animals were injected with DMSO. And this is the effect with aspirin. Both dinoaspirin and ipanoaspirin significantly reduced the tumor size, the primary tumor size. And this, we measured the tumor size as well, and this is basically the quantified data from the fluorescence of the GFP tag. And as you can see here, it showed significant decrease. We also looked at the effect of these compounds on metastasis by imaging the brain, but we did not see a significant effect. So the other aspect, as I mentioned, is to completely inhibit NOS2. So another postdoc, Julie Heinecke, in our lab, studied the effect of aminoguanidine, which is a NOS2 inhibitor, on using the same model. And she found that aminoguanidine decreases the tumor volume as well. And she also found that aminoguanidine significantly decreased the metastasis to the brain. So we thought this was really interesting because we did not see that with our NO donating molecules. Recently, Dr. Chenny Cheng's group in MD Anderson has also verified these studies, and she has done it with L-NAME, and she also found similar results. And they are currently planning to take these in for clinical trials. So they are going to start a clinical trial on these. So so apart from the molecules discovered in our group, I'm going to go over a few other molecules which have been studied by different groups. So this molecule, JSK, which was again developed at NCI Frederick in Dr. Kiefer's group, shows significant decrease of tumor volume in different cancer cell lines. They have studied it in breast cancer, melanoma, and in all of them, they saw decrease in tumor volume. This molecule releases nitric oxide in the presence of glutathione transferase, which leads to cleavage of this bond here, and then this part releases 2 equivalents of nitric oxide. Some of the other molecules which have been developed are in Dr. Sconfish group in North Carolina. He used these polymeric diazinium dilate-based molecules and showed that these can act as antibacterial agent. Another group showed that when a stent is coated with diazinium dilate releasing NO-releasing diazinium dilate, it does not form a scar tissue around it, and it does not block the stent. So these implants were much more effective, and it did not get blocked compared to the control, the non-coated version. Then recently, another group synthesized this molecule. So this is a macromolecule where the diazinium dilate is loaded within a calcium phosphate coated surface, and the surface is coated by hydrogen generator. So on shining light, this activates production of production of proton, which reacts with this calcium phosphate. And on this reaction, calcium phosphate will form a calcium hydroxide, which would dissolve in the presence of water in physiological condition, and diazinium dilate will be released. So then it releases, then the diazinium dilate decomposes to form nitric oxide. They studied this molecule in looking at the wound healing in corona, and they found that compared to the control group, these coated particles led to an increased wound healing in a short time, as short as 2 days. So I mentioned this ruthenium complex earlier. So this study was done by one of our collaborators in collaboration with our group. And this ruthenium molecule is activated by thiols to release HNO. And Lisa in our lab looked at the anti-angiogenic effect of this molecule, and she also looked at inhibition of NO so NO increases HIF stabilization, and when this molecule was added, it leads to inhibition of HIF-1 alpha. So the other class of molecule which has been used as HNO donors is cobalamin. So this is also a recent study in Angiochem where they showed that cobalamins can be used as HNO donating molecules. And as I mentioned earlier, HNO releasing molecules cyanamide and hydroxylamine is already in the clinic in some parts of the world. And cyanamide is used in clinic as an alcohol deterrent reagent. And hydroxylamine is used for treatment of sickle cell anemia. Also, I wanted to point out that like uh, there are many startup companies which have been built around NO and HNO research, and I'm not just saying that these molecules are important because we have patents on it. And so I came across this company, which is a really new company, and they work on NO donors based on solid support, and they can control the rate from fast donating to slow donating molecules. And they use these molecules for different viral and microbial diseases. And they have many molecules in phase 2 and phase 3 trials for treatment of herpes and other diseases. This company, Cardiomyocyl, was based on HNO research, and this they studied the effect of HNO in heart-related ailments. And this company was recently, last year, bought by Bristol Myers, and they are further developing these molecules. And right now, it is in the phase 3 clinical trial. And both of these are pharmaceutical company, and Jindo, I think. So this is a company which is based in Rockville, and they mainly work on development of research technology. So they are developing methods for detection of SNO compounds and other research devices for studying and characterization of different protein modifications by NO and HNO. So in conclusion, I would like to point out that donor compounds are really important in this field because for both NO and HNO, because not only from research point of view, but also from therapeutic point of view, because we want to control the levels of these biomolecules released. As for NO, the main chemistry is based on ferrous and ferric mediated heme chemistry and thiol chemistry. And for NO, it is mainly ferrous heme chemistry and radical chemistry. And these lead to different biological effects of both NO and HNO. And at the end, I would like to thank my advisor, Dr. David Vink, for giving me this opportunity, and Dr. Terry Moody for inviting me to give this talk. I would also like to thank Lisa Ridner and Robert Cheng, who are staff scientists in our group, and they helped me with many of the animal experiment and setting up many assays. Madison Greer is a current post-bac student who's working with me, and Veena Somasundaram is a postdoctoral student who is working on other projects which I did not talk about today. Also, these compounds that I talked about were synthesized in University of Arizona during my PhD, and during that time, I was a pre-doctoral fellow at NIH, and I did the biological studies here. And I would also like to thank one of our collaborators, Sharon, who was instrumental in analyzing the patient data. And thank you all for your time, and thank you for listening, and I'm open to any questions.
Debra, so am I understanding the chemistry? So the nonoids will donate HNO, but in the process, they always also donate NO?
Yes.
So it's really a combined approach in each one of those, or so are you able to dissect out if it's actually a synergistic effect between NO and HNO as opposed to effects of HNO alone?
So mainly we would quench the HNO with thiols and see how much it's producing, what's the ratio of NO and HNO. And we have found that the ratio varies. Like I showed you that CPNO ACOM molecule. So when it's protected, it completely produces HNO. So it it is a pure HNO donor at that point. Even though that has a shorter half-life, it is only 21 minutes in serum-free condition. But some of these, like ipanoaspirin, has a much longer half-life, and then ipanoindomethacin is even longer. And they, like, when we did the experiment under fetal bovine serum, they do not fall apart. The esterase bond is not cleaved, so they are still producing HNO in the media, like pure HNO. But in the presence of guinea pig serum, it will be cleaved and it would produce both NO and HNO. So it really depends on the conditions. In the cell, I mean, I'm guessing we don't really have we don't have the methodology to say how much portion is like NO. We do know that it is producing NO because we have looked with DAF assay, and DAF shows a much higher fluorescence for HNO, like it's 5 times high compared to NO. So we do know that it is producing HNO in the cell, but we do not know how much is the ratio. And like the decomposition process depends on like specific esterases. So it's not like cleaved by all esterases. So we haven't looked into that aspect as well.
Right, it'd be very cool if you could tune it so that you knew at a, for a side effect issue, so if you knew at an inflammatory site or within a malignancy, you get one ratio, and outside of that, you would get a different ratio that was perhaps benign, or some kind of activated by disease sort of drug.
But I mean, we thought it was still really cool because for some reason it is not toxic to it is not toxic to HUVEX. It is not toxic to MCF10A cells. I mean, like both Dino and ipanoaspirin. So.
Thank you.